Hi everyone, and welcome to the last of our lectures, which will focus on nitrogen metabolism and then cell signaling. Now, the first thing I want to go over is an overview of nitrogen metabolism. There are three terms or three processes that I want you to be aware of. Be aware of the terminology and exactly what's going on. So the first one is nitrogen fixation. So you can see that here in this small diagram and also on a larger scale in this diagram here. Nitrogen fixation is when molecular nitrogen from the atmosphere is taken in by things like nitrogen fixing bacteria and converted to ammonia. That ammonia then undergoes nitrification, which converts it to nitrates that can be absorbed into root systems, for instance. Since obtaining nitrogen is very, very important for cells. Nitrogen allows cells to build two main things. They are amino acids and nucleic acids. So without nitrogen, you would not have proteins and you would not have DNA or RNA built within the cells. There's also denitrification because when it comes to molecules and various substances, it's important to be able to cycle them throughout systems. And so denitrification will allow for putting back molecular nitrogen into the atmosphere by breaking down those nitrates or nitrites, okay, that were the product of nitrification. Okay, so all three of these end up cycling in order to properly get nitrogen throughout a system. Okay, so the first of those processes that I want to focus in a little more on is nitrogen fixation. And I'm going to have a recap slide next of the notes you should have on this. Now, as we mentioned before, nitrogen fixation is the reduction of atmospheric nitrogen to ammonia, okay? And then that ammonia will then go into nitrification. Now, when we talk about nitrogen fixation, I want you to remember the organism that actually accomplishes this is nitrogen-fixing bacteria, and you find them in these little nodules that collect on the roots of plants, specifically leguminous plants a lot of times, so things like beans and alfalfa sprouts. And so these nodules full of nitrogen-fixing bacteria on plant roots are very important because those plants and animals, including us, cannot fix nitrogen. Okay, they can't carry out nitrogen fixation. So they rely on the symbiotic relationship with these bacteria in those nodules. Okay, so that's very important to remember. Plants and animals cannot do nitrogen fixation. Instead, it's the bacteria in these nodules on the roots of plants. Now, what's actually required for this to happen, some of the terminology that you see in this figure here. So there's what's called the nitrogenase enzyme complex. Okay, the nitrogenase enzyme complex. It includes ferredoxin, dinitrogenase reductase, and dinitrogenase, okay? And these three work together in order to accomplish the reduction of nitrogen. So basically you have ferredoxin, the flow of the electrons from ferredoxin first, then you have the electrons flowing to the dinitrogenase reductase, and finally to the dinitrogenase. Okay, and the reaction for the reduction that's occurring is shown here. As you can see, it's an 8-electron reduction occurring, and when you have this, this is an important way to obtain ammonia, which can then go toward nitrification. And so that was a lot of information in a short time with a lot of terminology, so this is a recap slide for you to pause and jot down the notes from the previous slide. Now, when we talk about nitrogen metabolism, remember I said nitrogen is critical in cells for two main things, for building amino acids and for building nucleic acids. So one of the things we're going to talk about is amino acid biosynthesis and the control of that synthesis. And as you can see here, the control of that synthesis might look kind of complex, but in actuality, it's one main process that's occurring. It's feedback inhibition, which we've talked about before. That's a really efficient way for cells to inhibit things, okay? So the products can inhibit what comes before it on the pathway. Now, the example here is the inhibition of glutamine synthase, and it looks very complex because it has 9 allosteric inhibitors involved here. So all of these little, stop marks here are the 9 inhibitors. So serine, histidine, tryptophan, glucosamine 6-phosphate, CTP, AMP, carbamoyl phosphate, alanine, and glycine are all working as allosteric inhibitors to this glutamine synthase pathway. Okay. And that's an enzyme that's very important in amino acid synthesis, which you'll see a little bit later on. And now overall, even though this looks very complex, feedback inhibition is very effective because any one of those single product molecules can inhibit an enzyme that might be capable of synthesizing hundreds and thousands of product molecules. So you have that one product stopping the production of all kinds of products, and it really saves resources and energy. Now, when it comes to amino acid biosynthesis, the three main processes that you see occur are reductive amination, amidation, and transamination. Now, the textbook does not go into the details of each of these, so you're not responsible for knowing exactly what occurs in these reactions. I just want you to be able to recognize that these three terms are associated with how amino acids are synthesized. Now, for instance, the production of glutamate occurs by reductive amination, okay? And that's this figure here, whereas the production of glutamine is considered amidation, okay? And so basically, you have a lot of different processes going into the ultimate creation of amino acids, but they all rely on having nitrogen available. Now, we mentioned transamination, and transamination reactions are really important because they're basically the transfer of an amine group from an amino acid. So, for instance, here you see an amine group from an amino acid to an alpha keto acid. And what you notice is once that transfer has occurred, that alpha keto acid now has the amine group and thus is now a different amino acid from our original one. Okay, so in the example here, it shows glutamate becoming aspartate. And as you can see, it's not actually that the glutamate molecule is becoming the aspartate. It's that the glutamate is transferring the amine group to an alpha keto acid, and that alpha keto acid is what's now the new amino acid because it's taken on that amine group. Okay, now these reactions, and as you can see in the examples here, can be catalyzed by different enzymes. One example is glutamate synthase and glutamate dehydrogenase. And these are important, especially in plants, because they basically allow plants to cope with conditions of limited nitrogen availability because they can catalyze the transamination reactions during times when, they need a nitrogen source. Okay, so for instance, when nitrogen is limiting, the conversion of glutamate to glutamine. So the figure over here. Okay, so glutamate to glutamine, that can be a preferred mode of nitrogen assimilation. The supply of glutamate ends up depleted unless there's some way to basically restore it again. And so then the reductive amination of an alpha ketoglutamate, so alpha Kg in these figures, can then be a way to replenish. Okay, because these reactions, these transamination reactions, are reversible, and so you can go back and forth, as you can see in this figure and this figure. Okay, so it's a nice way to cycle nitrogen when it's limited. As promised, here is a recap of the key points that I want you to have in your notes. Now, in addition to transamination, what's called one-carbon transfers are also important in amino acid biosynthesis. Okay, and so an example of converting serine to glycine through a one-carbon transfer is seen here. And there are 3 important carriers to know as examples of carriers for these one-carbon transfers. We have biotin, tetrahydrofolate, and S-adenosylmethionine. Okay, and each one is a carrier of a particular group. So biotin will be a carrier of CO2. Tetrahydrofolate, which is in the example here, is a carrier of methylene and formyl groups. And then S-adenosylmethionine is a carrier of methyl groups. Okay, so each one has its ability to transfer one carbon units of, from one amino acid to make another one, and they each have a particular function or role. Now, unfortunately, even though we keep mentioning amino acid biosynthesis, not all amino acids can be synthesized in every organism. So even though E. coli is lucky in that it can synthesize all the amino acids they need, other organisms like us can't do that. And so any of the amino acids that we need but we cannot synthesize and we need to instead take in through our diet, these are called essential amino acids. And getting all of your amino acids and getting the proper amount of protein is a very difficult task. And we're going to talk more about that at the end of this lecture. But for now, focus on how we have what's called essential and non-essential amino acids. The essential amino acids are the ones that we can't synthesize and need to ingest. Okay, now, like I said, for a lot of people, this is not easy to do. And so a lot of people end up with protein deficiencies. And the unfortunate example that is very rampant in third world countries is seen in the pictures here. That's kwashiorkor. Okay, and you can tell when it's kwashiorkor because of that distinctive bloated abdomen. So you see that in a lot of the children in third world countries. And part of that is from the fluid retention that severe protein deficiency can basically make. And so it's a form of severe malnutrition. There are other types of malnutrition that are rampant, like marasmus, but they don't end up with that kind of bloated stomach and fluid retention that you get from protein deficiency. And it's unfortunately a lot more common in children. especially when the mothers in these countries end up stopping breastfeeding the child too soon because they end up quickly pregnant with another child. And that's actually part of where it got its name from. It comes from a dialect where that means something similar to the sickness the baby gets when the new baby comes, because they're basically weaned off the milk too quickly, and so they don't end up getting the proper amount of protein. Now, a theme that constantly pops up in our lectures here and in any of the biology courses is that in science, just like in every part of life, to everything there is basically an opposite event that can occur. You know, you need that kind of balance. If you're going to create something, you need a way to also destroy it. And so when you have the anabolism or anabolic reactions leading to the creation of amino acids, you also need catabolism or the catabolic reactions leading to the breakdown of those amino acids, okay? Now, there's going to be a recap slide in a minute. The big thing for these reactions is the first step, which is transamination. Now, interestingly, we just mentioned transamination in the creation or the anabolism of amino acids. So don't get confused thinking, oh, it's just the complete opposite. You know, we've seen, the complete reverse reaction. We've kind of seen throughout other lectures that that's not always the case, but ultimately the end goal is opposite. You know, before we were building, now we're breaking things down, and you ultimately end up, you know, with transamination being important in both of these different events. Okay. Now, when you have the transamination occur, okay, you end up with a carbon skeleton left behind. And the fate of that carbon skeleton ultimately depends on what type of end product is being produced. So there's two general pathways to think about, and it's based on whether you're looking at a glucogenic amino acid, so those are the ones highlighted in red in this figure, or a ketogenic amino acid, which is highlighted in green. Okay, and then you have some cases of both also highlighted, and you know they're categorized based on what the end product goal is. Now, with the two pathways, if it's a glucogenic amino acid, then the carbon skeleton fate is ultimately going to yield pyruvate or oxaloacetate upon degradation. Now, oxaloacetate we've heard before, so you know that this is important, being able to break these guys down into oxaloacetate, because that had a central role in the citric acid cycle. And, you know, it's a major starting point also for the production of glucose by gluconeogenesis. Okay, so that can be valuable to a cell. And then a ketogenic amino acid, what happens with them is the ketogenic amino acid is broken down to acetyl-CoA or acetoacyl-CoA. And that's important, you know, because that can ultimately lead to the formation of ketone bodies. And because we've seen acetyl-CoA is a major player in terms of heading into a lot of important cycles, also including this citric acid cycle that we mentioned a minute ago, you know, oxaloacetate being important there too. Okay, so ultimately those are the two main pathways, is that you can either have the glucogenic pathway or the ketogenic pathway, and you're either producing pyruvate or oxaloacetate with the glucogenic pathway, or you're producing acetyl-CoA with the ketogenic pathway. And so this is a recap for you to pause and jot down the notes from the previous slide. So now with the anabolism and catabolism, or the synthesis and breakdown of amino acids, it's important to consider, well, what happens to the excess nitrogen that's going to be excreted during these kinds of processes? So there are three forms in which excess nitrogen can be excreted, and I want you to know each one, what's going to be excreting it, and why that organism chooses that way. So the first one is ammonia, and ammonia is going to be the most toxic and concentrated of these forms. Now this one will be excreted by fish because if you think about it, these fish are in a water environment. So that water environment helps dilute down the toxic ammonia and protects the animals from what's being excreted. Okay, and that one, when you look at this here, you'll see that ammonia is the form that requires the most water and the least energy, and that's perfect for fish because they have a lot of water available. Next up, you have urea, and that's the form of excess nitrogen excretion that we use and other terrestrial or land animals use. And as you can see in this figure here, it's the perfect balance, the intermediate when it comes to water needed and energy needed. Because, if you're a human, you got plenty of other things to worry about. You don't want to have the reaction that's going to require the most water or the most energy to deal with it. And it's not going to be toxic to us the way that ammonia would be. Okay, so we have urea. And then lastly, birds and other reptiles, they use uric acid as their way of excreting excess nitrogen. And what's good for them about this one is you can see here, there's not water, not an excess of water. And that's because this one is a water insoluble waste, okay? Whereas things like urea is water soluble. So by having a water insoluble compound, these birds are not going to build up excess water while they're forming this excretion product. And so that allows them to avoid the excess water weight while flying that might weigh them down or hinder their ability to properly fly. Okay, so this is a nice little summary over here to help you visualize, the one with lots of water and then the one with not so much water, and then the perfect balance one. So now that brings us to the urea cycle, and I'm not going to make you memorize all of this. I don't, want you to know how to draw out the structures or anything. I just want you to know that this urea cycle does exist and that its main function is to eliminate toxic ammonia from the body. And so, that's like I was just saying in the previous slide, we convert any excess nitrogen and ammonia into urea because it's a way for us to avoid having exposure to the toxic effects that ammonia would have on us. So, unlike fish that could just, swim around in water and dilute the ammonia, we convert it or eliminate the issue by creating urea. Okay? So it's very important pathway that ultimately leads to the excretion of waste products of nitrogen metabolism, especially those of amino acids. So now, like I said earlier, nitrogen, I always use the trick in my intro level bio classes or in microbiology when we're going over what each of the chemical requirements are and what, things like nitrogen and sulfur and carbon are used to create. I use the memory trick of NaN and NaN to remind us that nitrogen is to make amino acids and nucleic acids. So we already talked about the amino acid anabolism and catabolism. So now I want to take a minute to go over the nucleic acid side of things. So here we have an example showing you purine nucleotide biosynthesis, okay? And when we look at this, I'm not going to make you memorize any of the components or the enzymes or anything. I just wanted you to see what can happen in terms of AMP synthesis and GMP synthesis. And ultimately, when you have these kinds of synthesis in terms of purine nucleotide synthesis, the growing ring system is bonded to the ribose phosphate while this whole purine skeleton is being assembled. And that'll come back in a minute as a difference when we look at pyrimidines. Now, when you have this, this synthesis, the carbons of the carbons that you see of the rings here, they come from different sources. They're from different derivatives. And so this figure shows you each of the components of this ring structure and where they're coming from. Okay, and then you see here the attached sugar group to the ring structure. Now, just like a lot of the slides we go over in biochemistry, the answer to this slide in terms of purine nucleotide biosynthesis, how it's being controlled, is the same as for a lot of the things we've talked about. The answer is feedback inhibition. And like I mentioned earlier, feedback inhibition, which you see by all of these dotted arrows, these dotted lines showing how a product goes back to be an inhibitor to the previous enzyme or the previous step enzyme. You see over and over all of these guys being inhibitors. Like I mentioned earlier, feedback inhibition is very, very efficient and effective for cells. You can have this one product, so for instance here, GMP, that one product, then going back and being an inhibitor that stops a lot of future steps from ever being able to happen by stopping an early enzyme in a pathway. So you have one molecule being able to prevent the expenditure of and waste of so many resources and energy to create, a lot of different molecules. Okay, so feedback inhibition is how purine nucleotide biosynthesis is controlled, and it's a very effective, efficient means of inhibition. Okay, so like I keep emphasizing, the idea that you always have to think about the opposite things in biology and in life. If you have the building or biosynthesis of purines and that control, then you also have to think about the breakdown of these molecules. And so I'm going to have a recap slide on the next page so that you can see exactly what is going on, because it's a little tough to follow sometimes. When you have the breakdown of these purines, you first have hydrolysis to the nucleoside, and then next up is the free base to get further degraded. And so there's a pathway of key molecules that I want you to be aware of. So for instance, when you see xanthine, to know that that is part of the purine catabolism process, because basically you end up with the deamination of guanine to xanthine, which you see over here, and that produces hypoxanthine. And the hypoxanthine is basically going to be the base corresponding to the nucleotide inosine, okay, which you have over here. And so you can see, while you have this pathway kind of showing here, you at the same time see this pathway going this way, and basically that hypoxanthine can be oxidized to xanthine. okay? Because, like I said, with everything, you have kind of like the reverse options available. Now, what's important here is xanthine. The reason why I keep saying that name and emphasizing that I want you to know that it's part of this pathway, because I'll never make you, memorize the chemical structures and the exact pathway of this goes to this, goes to this type of thing. But I want you to know xanthine is part of purine catabolism, because that's the one that then gets oxidized, as you see over here, to turn it into uric acid. And we've mentioned uric acid now in this lecture because uric acid is what birds and reptiles use as their end product to then excrete, okay? Because we're still on, nitrogen processes and nitrogen metabolism here. So xanthine gets oxidized to uric acid, and this can be, excreted in terms of, as the final product in things like birds and reptiles. And then in all other terrestrial animals, like that, which includes mammals, all the other mammals, you get the conversion to allantoate, which is then degraded to, oh, they don't have it over here. Oh, they do have it over here. Glyoxylate, and these guys end up producing urea, okay? Because we've mentioned urea is another form of excretion in terms of these metabolic pathways. Now, what's interesting is if during these process of purine breakdown, if too much uric acid builds up rather than, being able to be excreted or converted, if too much uric acid builds up, then humans can get what's called gout. And gout is a human disease where you have the overproduction of uric acid, and so you get deposits of that uric acid in your joints, especially in your hands and feet. Okay, and that's, it's barely soluble in water, and it just collects. And ultimately, there's something called allopurinol. So because this is purine catabolism creating that uric acid causing this gout, the treatment medication that they made is allopurinol. Okay, and that basically works by inhibiting the degradation of this hypoxanthine to xanthine, which, as we just said here, is what gets oxidized to uric acid. Okay, so if you block the hypoxanthine with allopurinol, you will block the conversion to xanthine, which blocks the production of uric acid and prevents that excess uric acid from building up in joints of the hands and feet, which can be very brutally painful for people if you've known anyone who's had gout. Okay, and then they have to avoid certain foods as well to kind of reduce this build up. Avoid foods that are high in purine content, such as, shellfish. Okay, there's another term that you see pop up when you study purine catabolism that I wanted you to be aware of, which is salvage reactions, and that's basically salvaging or reusing compounds such as purines that basically require a large amount of energy to produce. So rather than producing them from scratch, you're basically recycling and using these energy intensive compounds. And since that was a whole lot of information at once, here's a recap slide. Make sure you really know, xanthine and the connection to gout with the production of uric acid, and that allopurinol, circle star, highlight that term, is used to treat gout by inhibiting the degradation of that hypoxanthine to xanthine to uric acid pathway. Okay. Now, since the exam is coming up so quickly, since this is the last of the exams, I will include, more recaps in this one. So there's going to be a recap slide on these notes in a moment. Since we covered purines, it's now time to take a minute just to look quickly at pyrimidine biosynthesis and catabolism. Okay, so the overall um, pyrimidine biosynthesis is different from purine nucleotide in that, remember I mentioned before, the idea of the ring being assembled to the ribose would be different. So in the case of pyrimidine biosynthesis, that pyrimidine ring is assembled before it's attached to the ribose 5-phosphate, whereas in purine synthesis, you saw that the growing ring system was bonded to the ribose while the whole skeleton was being assembled. Okay, so in purine biosynthesis, the attachment to the ribose and the ring is at the same time as the overall synthesis, whereas in pyrimidines, the ring is assembled before it's attached to the ribose. And what happens in pyrimidine biosynthesis is that the pyrimidine ring is coming from what's called carbamoyl phosphate and aspartate. So if you look here, carbamoyl phosphate and aspartate, so these two here are leading to the ultimate production of the ring structures. Okay, and then for catabolism, the pyrimidine nucleotides are broken down first to the nucleoside and then to the base, just like, the purine nucleotides are. So this is a much quicker slide because overall it's a very similar idea to what happens with purines. And as promised, here's the recap of the main points so you can pause and jot these notes down. So now, when we keep mentioning nucleic acids and, purines, pyrimidines, we keep mentioning ribose and focusing on that, which means, RNA and ribonucleotides. But as you know, deoxyribonucleotides are crazy important too. That is, my all-time favorite molecule is DNA. So the conversion of ribonucleotides to deoxyribonucleotides is very important. And ultimately, when you have this conversion, I'm going to have a recap on the next slide. Like I said, there's a lot more recaps in this lecture for you to help you as you prepare for the exam so quickly. When we look at this conversion, first of all, it's catalyzed by, so I'm trying to find it in the figure here, thioredoxin reductase, okay, thioredoxin reductase, and ribonucleotide reductase, okay? So they highlighted in pink um, the main enzymes involved in this process, and circle star highlight ribonucleotide reductase. That's going to be the big main one. And what happens in terms of this conversion is that the ribonucleoside diphosphates are reduced to, I'm trying to see if it's in here, um, in the figure, they're reduced to 2-deoxyribonucleoside diphosphates in all organisms with NADPH as the reducing agent. And then in this process, not only do you need to have this enzyme present here, but you also need to have, as you can see in this figure, the thioredoxin and the thioredoxin reductase. Okay, so I said circle star highlight because this is the main guy that people associate with this process, the ribonucleotide reductase. But you can't have this process happen without the NADPH that you're seeing and the thioredoxin with the thioredoxin uh, reductase enzyme. And ultimately, the thioredoxin and the thioredoxin reductase, they are going to be basically part of the electron carriers and the intermediates in this process. And the thioredoxin will contain a disulfide group in its oxidized form and two sulfhydryl groups in its reduced form. And the NADPH reduces the thioredoxin by having the enzyme thioredoxin reductase catalyze the process, and in turn basically reduces a ribonucleoside diphosphate to a deoxyribonucleoside diphosphate. Okay, now you don't have to know all of the details in terms of this process, but just be aware when you see these terminologies, these three main words here, the ribonucleotide reductase and thioredoxin, as well as that reductase there. I just want you to be aware that these are involved in this process, okay? Because I'm not worried about you being able to draw out processes and whatnot. I care about you being able to recognize terminology. And since that was a bit complex in terms of me saying a whole lot of words, here is the recap for you to pause and write down some notes. So the last thing that I want to mention with regard to nucleic acids is the conversion of DUDP to DTTP. Okay, so uracil to thymine. And I'm not going to make you memorize all of the steps to it or all of the conversion. I'm going to have the key points in a recap slide next. I'm mainly going to focus in terms of what I want you to know in relation to cancer treatment. But for this slide, I do want to point out that a 1-carbon transfer is required for this process. So that's a term that you heard in previous slides. Ultimately, the reactions that you're going to see occurring here include the thymidylate synthase reaction, okay? And you can see the source of the 1-carbon transfer, and they highlight in pink that the 1-carbon that's basically getting moved around and converted in the process. And you can see that the 1-carbon carrier is over here, THF, so tetrahydrofolic acid or tetrahydrofolate. And that's going to be, a key player in this process. And the process, you'll also notice, requires NADPH and this enzyme here, dihydrofolate reductase. But the key point that I really want to make is in relation to cancer cancer treatment. Because when you look at this conversion, ultimately having a supply of DTTP is critical for cells in terms of DNA synthesis, okay? Because as you know, uracil is found in RNA and thymine is required in DNA instead. And so cells need DTTP for DNA synthesis. So if you inhibit the enzymes that catalyze the production of this thymine, you're going to inhibit the growth of rapidly dividing cells. And now when you think of rapidly dividing cells, the one word that should come right to mind is cancer, because that's what cancer is. It's uncontrolled cell growth and division. So it's rapidly dividing cells. So cancer cells, because they're so fast growing and replicating and dividing, they depend on just a continuous DNA synthesis. And that means that they depend on the continuous conversion and the continuous production of this thymine, the DTTP, right? And so inhibitors of things like the thymidylate synthase that's involved in producing or converting to the DTTP, if you inhibit enzymes like that, which can be done with things like fluorouracil, or if you inhibit this guy here, the dihydrofolate reductase, so circle star highlight these guys here, the thymidylate synthase and dihydrofolate reductase, if you inhibit those enzymes, you inhibit the conversion of DUDP, or you can uracil to thymine, right, which inhibits DNA DNA synthesis and the ability for cancer cells to be rapidly dividing. Okay, and so that's been a target now in terms of cancer chemotherapy, and ultimately, it'll mainly hurt cancer cells because they're undergoing this conversion and replication and division so much faster than the normal cells. So it can still harm the normal cells, but more to a minimal degree because they're much slower in terms of this process. Now, as I promised, here's a recap slide. And like I said in the previous slide, you don't have to memorize all the steps or anything in terms of the conversion. I want you to really focus on how this conversion can be used clinically in terms of cancer treatment options. Okay, so now we head into the final chapter. Yay! And this chapter will basically integrate metabolism to cell signaling. Now, the first thing I want to show in this section is how all of the metabolic pathways are connected, so much of what we've talked about. And as you can see, the big shot main connection is that all forms of metabolism can ultimately be linked to photosynthesis. So circle, star, highlight photosynthesis and the energy that it's getting from the sun. Okay, because if you think about it, the light reactions that we talked about in photosynthesis, they make ATP and NADPH, right? And then those are then used in the dark reactions, okay, the carbon dioxide fixation reactions, to produce carbohydrates. And as you saw throughout this course, carbohydrates are then a main fixture in terms of being nutrients for various other organisms. And the aspects of this process, ATP, NADPH, carbohydrates, they all feed into catabolism, anabolism, all of the different processes that we talked about. Okay, so they end up being a big direct link to catabolism and anabolism. And you also see some other molecules being, big time links for the various processes. Things like PEP, pyruvate, and acetyl-CoA, for instance, we saw constantly coming up in different processes we talked about. Okay, so there's a lot of connections when you think of metabolism. This is just a recap in terms of the main points of the previous slide for you to jot down in your notes. Now, in the previous slides, when we talk about metabolism and all the connections that exist, one of the cycles that you see constantly connected in terms of the other metabolic processes is the citric acid cycle. And so this slide just summarizes for you the three main points that make citric acid cycles so critical to the study of metabolism. The first one is that citric acid cycle is connected to the catabolism of nutrients, specifically carbs, lipids, and proteins. On the opposite side of that spectrum, in addition to catabolism, the citric acid cycle is also connected to anabolism, okay? And that's the building of sugars, lipids, and amino acids. And then lastly, it's connected to the individual metabolic pathways as well. Okay, so you kept seeing citric acid cycle coming up quite a bit throughout this course. And so this slide is just to summarize some of its significance. So now I just want to take a moment to deviate to a biochemical connection, a clinical health example, just to show you how biochemistry can play a role in something you may not think of it being connected to, which is alcoholics and any kind of use of alcohol. So the more alcohol you drink, and I'm going to have a recap on the next slide, the more alcohol you drink, the more tolerant you become to more and more or higher and higher blood alcohol levels, levels of blood alcohol that could kill someone who's not used to drinking. And the reason that is that the enzyme responsible for breaking down the alcohol dehydrogenase, it's an inducible enzyme. Okay, so its level increases in response to alcohol use. Okay, so the more you use alcohol, the higher the levels of this enzyme is going to be. And when you look at how this enzyme actually works, the first reaction that it involves, it happens very quickly in alcoholics, okay? So an alcoholic's very quickly able to, break down the intoxicating effect of alcohol by, breaking down that alcohol quicker. And so they can tolerate a higher blood alcohol level. Whereas for everyone then, including the alcoholics, the second reaction is what's considered rate limiting. And it includes a byproduct of acetaldehyde. And with that, that causes headaches, nausea, and things like hangovers. Okay, so you end up having the problems that everyone else has when they drink too much. Now, and when the body no longer has the alcohol. Now, another thing, in addition to hangovers and things like alcohol poisoning and drunk driver accidents and all the bad stuff that alcohol can do to you, people don't realize that alcohol is also going to put, weight around your stomach. Okay, so I've seen a lot of people, they'll be dieting and then they'll be, oh, I go to the gym, I diet, this, that, the other thing. And then they go to the club or the bar and they drink like crazy, not realizing that they are making their body and even their face all kinds of hideous and noticeably bloating and everything, because alcohol is basically empty calories. And malnutrition ends up being very common in alcoholics because most of the time what they're intaking is just alcohol, and it may fill them up because it's liquid taking up room in their digestive tract, but it's not giving them any important nutrients or any vitamins. Ethanol is also what we call a teratogen, so that word there, and teratogen means that it causes the malformation of embryos. And there is no safe limit in terms of women who are pregnant having alcohol. The slightest amount is a teratogen and can cause malformation. And you see in this figure here, a lot of children born from an alcoholic mother end up having not only malformations in their physical features and defects there, but major defects in terms of their CNS, their central nervous system. So mental retardation is a big time consequence if the baby even ends up being born, because it can have toxic effects too within the development where you may not get the baby born. Okay. Now, what's interesting in terms of alcohol use is I mentioned hangovers. And the reason we end up having hangovers is that when you use things like alcohol or any kind of drugs, a lot of them are basically structural analogs of things like serotonin and epinephrine. And what this does to your nervous system is the use of these drugs and alcohols can cause the release or the production of endorphins and enkephalins. And these are basically the enkephalins you may not have heard of as much as you hear endorphins. They cause, happiness because enkephalins, they are basically the brain's own opiate painkillers. So when you have this painkiller, it's, a pleasant effect. But the thing is, when you drink the alcohol, ethanol is inhibiting the natural synthesis of enkephalins. Okay, so you end up with this lack of enkephalins after you've been drinking all of that alcohol. And so that's what the pain that you end up feeling in terms of a hangover, because by that point your body has too low of the enkephalins. Okay. Now, this is all summarized here in this recap slide, so you can pause it and jot down your notes. So now let's connect biochemistry and some of the terminology we've learned to nutrition, which is very significant in your everyday life. So first off, I want to point out that we are heterotrophs as opposed to autotrophs. So we need to eat or ingest organic matter for carbon, whereas autotrophic organisms are self-feeding. They can basically use carbon dioxide and inorganic matter to obtain their carbon. This is significant. You need to get carbon because carbon is then used to make all four biomolecules. So cells need it to make lipids, proteins, carbs, and nucleic acids. Now, when we talk about nutrition, you'll see catabolism of macronutrients come up a lot. That is basically the breakdown of carbs, fats, and proteins for energy. But the big one that I want you to circle, star, highlight is protein, because protein deficiency is a major concern in nutrition. And we saw that in previous slides when we saw the malnutrition effects and the bloated bellies of those poor children who were suffering from protein deficiency. Now, protein is such a big issue in terms of nutrition because it's not as readily available as the other nutrients, and unlike things like carbs that have carbs and lipids that have stored forms where you can take in some at one moment and then store some for when you need it later, proteins do not have a storage form. So you need to be taking in your essential amino acids every day in a proper quantity. Otherwise, your body's not going to be able to undergo protein synthesis. And since proteins at the same time are constantly being degraded, you really need to balance out and be able to intake the proteins and the amino acids that you need. Now, in addition to those macronutrients, meaning those nutrients that you need in a large amount, which are the carbs, lipids, and proteins, we also have micronutrients, which are vitamins and minerals. And so these you need on a daily basis in smaller amounts than the macronutrients. Now, the reason why these are both so significant is you've heard in our previous lectures that vitamins are coenzymes and minerals are cofactors, and both coenzymes and cofactors are critical in terms of enzyme function. You can't have enzymes guiding all of metabolism if they do not have the vitamins and minerals to properly activate them and help them to function. Now, when it comes to vitamins, keep in mind, like we discussed previously, there are the fat-soluble vitamins of vitamins A, D, and E, for instance, that you need to be very careful with because those you can actually overdose on and have, excess be toxic because they end up being dissolved and absorbed into fat and accumulate in your adipose tissue. Whereas the water-soluble ones like vitamin C, they can be excreted as they dissolve in water, okay? You also have minerals, and those are the inorganic substances, and they are required for a lot of different, life processes. The main ones, so the ones that we can call macro minerals, even though they're micronutrients, they're macro minerals in that out of the minerals, you need more of them compared to other minerals. Those include things like sodium, potassium, chloride, magnesium, phosphorus, and calcium, which are ones that I'm sure you hear on a daily basis and a lot of times see as electrolytes, for instance. Now, when it comes to nutrition, people used to rely on the old food pyramid, which you see here on the left. There were some issues with this one. For instance, it basically over-glorified simple carbs, and it villainized fats and oils and sweets and basically put all fats together as being bad rather than distinguishing the fact that, saturated fats are really bad, but there are monounsaturated fats that can be very helpful and healthy for you, as we've seen in other lectures. So that was a big problem in the old pyramid, the overemphasis on carbs and the villainization of fats. The new pyramid, the big thing I want to point out to you that is most significant and valuable in it is that it now emphasizes more than anything that exercise, being active, is very important when it comes to proper nutrition. It is not just about what you eat. There are so many people who will starve themselves rather than, really balance their diet better and add a lot more exercise and activity. 20 or 30 minutes of exercise each day can make a world of difference to your overall health, especially to your heart health and your mental health, including brain function. So it's very important to emphasize being active when it comes to nutrition as well. Now, unfortunately, when it comes to nutrition, America is known for not being all the greatest in terms of following proper nutrition. And there is a major crisis in America in terms of obesity. And, recent figures show more than 1/3 of the population is clinically obese, which is basically, when you look at the definition of obesity by the NIH, that means that all of those people have at least 20% more weight on them than is ideal for, let's say, their height, age, and gender. Okay. Now, a big contribution to this is, artificial sweeteners, fat substitutes, processed, foods, the things that are being added to foods, and, the fast-paced society that we now live in. More people going to work and people relying on things like takeout and junk food a lot. Soda has been a huge, huge problem in terms of the weight crisis. Now, we mentioned previously a protein that plays a major role in obesity, and you can see that in the figure here. I always love the poor little leptin deficient mice. They end up obese. And so basically leptin plays a major role in appetite and appetite suppression. So basically, when there's a mutation in the leptin gene, as they do with these guys, and they make it so that the receptors can't bind leptin, you end up having mice with increased appetite and decreased activity. So they're not moving around as much as they should, and they end up with a lot of weight gain. And then if you inject leptin into those same mice, they end up now decreasing their appetite and increasing their activity, which results in weight loss. So leptin is an appetite suppressant and can, thus block you from having obesity. Now, in addition to what you eat and how active you are, there is a daily activity that is very critical for proper metabolic rates and weight, and surprisingly, it is sleep. And they found that in studies looking at sleep, when you have just even a week of reduced sleep, the subjects in those studies ultimately began to show resistance to insulin, which ends up making them very similar to having type 2 diabetes. And when you have that kind of an issue, your levels of glucose in the blood then end up elevated and give almost a pre-diabetic state and drop your metabolic rate. So metabolic rates in those studies with just a week of sleep deprivation ended up 8% lower. And so if your metabolic rate is lower, you're going to be less active during the day, and you are going to end up, not being able to break down the food as quickly and ending up overweight or leading to obesity. So sleep is very, very important for proper health as well. So now, since this chapter focuses on metabolism and cell signaling, it's important to think about hormones and second messengers, because those are very important in terms of signals between cells and from outside the cell. And they're huge means of intercellular communication, basically. And this intercellular communication takes place thanks to the endocrine system, which are the ductless glands that produce hormones. And the hormones are basically intracellular messengers. And we're going to see in the next slides about the cascades and the messages that they can end up sending. Now, these hormones are very important to try and put the body at homeostasis, which is basically the balance of biological activities. So whenever you hear homeostasis, think equilibrium and balance within the body. Now, one of the other things I want to mention in terms of hormones is a particular example of hormones, and that is epinephrine and norepinephrine, which are responsible for your fight or flight response. Okay, and that's just an example of how hormones can basically trigger a response based on an external stimuli, such as, you seeing fire or you seeing a gunman standing in front of you. Now here I just listed out some terminology that you see with hormone or studying hormones, and I have a recap on the next slide with all of the definitions. The first one that we have here is hypoglycemia, and with hypoglycemia that just means low blood sugar. Then I wrote the hypothalamus, which is what will secrete a hormone releasing factor that basically stimulates the release of various hormones from endocrine glands, because it releases first trophic hormones, which are the hormones that act on a specific endocrine gland to trigger the release of hormones from those particular endocrine glands. So basically you could see that it's a pathway of signals to each other leading to hormones being produced. Then you have hypothyroidism, and hypothyroidism is basically because the thyroid is not producing enough of thyroxine, which is an iodine derivative hormone released by the thyroid. And if you end up with too much of thyroxine, instead you get hyperthyroidism. So if you have low thyroxine levels and you have hypothyroidism, you basically will see that present as having lethargy and obesity, whereas hyperthyroidism has the opposite effect. And the other clinical area that you end up with a lot of complications is based on the adrenocorticoids, and there are two categories of those. There are the glucocorticoids that are involved in carbohydrate metabolism and inflammatory responses, and then there are the mineralocorticoids that are involved in basically the water-salt balance and excretion from kidneys. Now, if the adrenal cortex is not functioning properly, you can either end up with Addison's disease or you could end up with Cushing's disease. And this one here shows Cushing disease characterized by, you see, this is how the boy originally looked, and then this is once Cushing took over and there was an imbalance in the adrenal corticoid levels. And so you can see characteristic is the major increase in adipose content. You basically hear it called moon face as one of the ways to identify Cushing's, and a lot of it's a whole lot of water retention in that kind of illness. The last example with hormones here that I give you is growth hormone. So GH stands for growth hormone. And if there's too much growth hormone, you could either end up with giantism or acromegaly. The difference between them is giantism, the excess amount of growth hormone is from birth, from the beginning. You see this major growth hormone amount. Whereas acromegaly, you can see this is all the same girl, but it doesn't develop until over the age of 30, where suddenly the imbalance of growth hormone takes hold and you see a major deformity in terms of the physical features, especially hands and facial features. And then lastly, if instead of being too much growth hormone, there's too little, you end up with dwarfism. And that one can actually be treated early on if they give basically injections of human growth hormone before the child reaches maturity. The last thing on here I have is second messengers, and they're basically the final part of this pathway. They bring about the changes in the cell as a result of the reactions with hormones. So it's basically a substance that gets produced or released by the cell in response to the hormone binding to a receptor. And so the second messenger is actually what will cause the final actual response that you see from that hormone. And since that was a whole lot of terminology that I wanted you to know the definitions of or significance, I have listed out the notes here for you. Now, when you hear the term second messengers, I want you to think of these three examples. So know that cyclic AMP, calcium ions, and receptor tyrosine kinases are all examples of second messenger systems in the cell. First up, cyclic AMP. That's important, especially in terms of stimulating CAMP or cyclic AMP dependent protein kinase, which then, regulates various activities in the cell, including transcription factors. And with this cyclic AMP, one thing I want to point out is it tends to interact with G proteins, and G proteins can be destroyed by the disease cholera. So cholera produces, a toxin that will destroy G proteins, and that basically leaves you with excess stimulation of adenylate cyclase and basically chronic elevation of CAMP levels. But the big problem with cholera, which is caused by Vibrio bacteria, is the patient ends up with severe diarrhea. And with diarrhea, you end up with dehydration. And so the big problem in terms of the dehydration and the illness side effects is that you have excess CAMP or excess, cyclic AMP, and that produces a large flow of sodium ions and water from the mucosal surface of epithelial cells into the various parts of the intestines. And, if the patient's losing a lot of fluid and salts and you can't replace that, it could be very, very dangerous for them. Calcium ions, what I want you to know for that is protein kinase C, which is a family of protein kinase enzymes, and they regulate metabolism. And what they end up doing is they phosphorylate certain enzymes, including the channel proteins that control the flow of calcium in and out of the cell. And by basically controlling that flow of calcium, they can, affect the reservoirs of calcium and the supply of calcium that a cell gets. And then the last one is the receptor tyrosine kinases, and that's what's in this figure here. And so as you can see, where are they found? Well, they span, they cross the cellular membranes. And the way that they work is that they're allosteric. So allosteric means a change in shape of a protein or an enzyme specifically. And so what happens is when the hormone binds to the region outside of the cell, you end up having the induction or inducing a conformational change in that tyrosine kinase domain. And that then activates the kinase activity. Okay, so the allosteric aspect is responsible for the activation of the kinase activity. Now, when it comes to hormones controlling carbohydrate metabolism, this is something we've already previously spoken about, so I'm not going to go into the details of this again. I just want you to be aware that the three hormones responsible or critical in carbohydrate regulation is epinephrine, glucagon, and insulin. And a lot of hormones, including these guys, work by signal cascades, which you can see in this figure here. Basically, what a cascade is the series of hormonal control or activations of hormones that ultimately amplifies. So if you have a small amount of hormone and it triggers a cascade, you end up amplifying the effect or the abilities of that hormone. So the last thing that I want to go over to finish this lecture and this course on is insulin, since it's something we've mentioned quite a few times and it is very significant, especially in terms of the clinical stuff and possibly in your life as well, since many people end up developing diabetes, which is the disease usually associated with insulin issues. Now, insulin is specifically a peptide hormone secreted by the pancreas, and the receptors that it uses are what we just mentioned before, tyrosine receptor kinases, which are those allosteric kinases that span, as you can see, span across the cellular membranes. Now, with the insulin, a big thing I want to point out is even though the mechanism is still ultimately being studied and constantly, being researched because of the issues in terms of diabetes and all, the way that it mainly seems to work is that insulin signaling causes the movement of what's called GLUT4, so the glucose transporter protein, GLUT4, and has it move from the intracellular vesicles to the cell membrane. And then once it's part of the cell membrane now, this protein is able to allow more glucose to enter the cell. And if the glucose is entering the cell, that means it's getting removed from the bloodstream. So it is lowering the blood sugar levels. Okay, and that's what insulin is most famous for. And, failure to have proper glucose transport is the main characteristic and the main risk associated with diabetes. Now, insulin keeps coming up as such a critical hormone, and one of the ways that we can demonstrate that is by listing out and looking at this table as well to see some of the various areas that it ends up affecting, because insulin can affect many different enzymes, and it's involved in glucose breakdown pathways, in the storage of glucose through promoting the synthesis of glycogen and reducing the breakdown of glycogen. It's involved in fatty acid synthesis and in cholesterol synthesis, triacylglycerol synthesis, so a lot of the different lipids it's involved in their processes. And this basically shows a whole bunch of the exact targets and what's going on that I also highlighted here. Okay, and that brings us to our very last point. We keep mentioning diabetes, so I just wanted to give you a quick breakdown of what is actually meant by diabetes. So there's type 1 and type 2. If someone has type 1 diabetes, that means they have it from birth. They were born with this issue, and it's basically that their body is not able to make insulin or doesn't make enough of it. And this is usually because their beta cells of the islets of Langerhans in the pancreas that are responsible for making insulin have been destroyed, usually by an autoimmune disease. So it starts off very early age, and the only way to treat this one is to give the person insulin. So for instance, insulin injections that have been made through recombinant DNA technology. Then there's type 2 diabetes, which has become rampant. It is highly on the rise. because of the obesity crisis that we mentioned a few slides back. And basically type 2 diabetes, which develops in adults, mainly, obese adults who have improper dietary habits. These bodies basically end up making a normal amount of insulin, but it ends up not having a sufficient effect. Okay, so it's not binding correctly to receptors, or it's not transmitting secondary messenger signals properly. Okay, and so again, this is the one that you associate with obesity and higher age in adults, and now it's even being linked to Alzheimer's as well. An area where you see insulin affecting, everyday life is also in athletes. And so when it comes to athletes, they have to be very careful about their diets, the day before their sports event and the morning of especially. And so, for instance, for breakfast, they have to be careful and choose something that is not a large carbohydrate-filled breakfast, because if they have a large amount of simple carbs in the morning, they'll have a major rise in blood glucose, and that'll cause insulin to rise, which will then cause their glucose to plummet and go below baseline. And that can be very dangerous for them, especially if they're in the middle of the sport when that happens. Now, also, insulin has been linked in terms of tumor cell proliferation, and for many years it was studied, breast cancer tumor cells were studied in labs, and when given large cocktails of glucose, insulin, and the growth factor EGF, the tumor cells would basically thrive on that cocktail, but when they were deprived of insulin, they would end up dying. And so it's seen insulin seems to be important for feeding tumor cells as well, and for basically, enhancing tumor cell proliferation. Okay. And I just want to point out, when I marked here what other diseases insulin now linked to, you can write Alzheimer's and cancers. So here is the last recap for today, because that was a lot of information on insulin and diabetes. You can pause and jot down your notes. And that is it, not just for today, but for this course. Thank you so much for your participation in the Remind app and in your participation in the exams. I know this has not been an easy semester for most students, and so I truly thank you. If you have any questions, day or night, please do not hesitate to ask me in the Remind app or through email. Thank you, and have a great day.